Hello, class. This week we're going to tackle probably one of the most popular chapters in exercise physiology, the chapter on skeletal muscle structure and function. So you see these terms used together a lot. Structure will refer to the anatomy, which is where we're going to start, looking at how a muscle is set up, what the structures are within it, etc. And then we'll transition to the function, which is the physiology, which is how it works. So the structure is what it is, function is how it works, structure is anatomy, function is the physiology. So just a couple sort of overall facts about what we call skeletal muscle. Now there are different types of muscle in the body. Smooth muscle is typically found in our organs and blood vessels. We do not have conscious control over smooth muscle, okay? Skeletal muscle are the muscles that move our bones, and we do have conscious control over skeletal muscle, okay? The other type of muscle is cardiac muscle. Cardiac muscle and skeletal muscle are kind of similar in their function, except of course we don't have conscious control over our cardiac muscle, okay? So with skeletal muscles, there are about 600 different muscles in the body, okay? Each muscle is wrapped in what we call connective tissue, and that connective tissue is what attaches the muscle to the bone, okay? Different types of connective tissue are tendons, ligaments, and then fascia, which is sort of the layer that wraps the muscle, and then ultimately fuses together into a tendon which attaches to the bone. It is through the connective tissue that we produce movement, because as the muscle contracts, that force is transmitted to the bone via the connective tissue, and that makes the bone move, okay? Muscle cells are a little bit different from normal body cells in that they have what you see here, a cylindrical shape. So rather than being round or circular like most body cells, they're cylindrical, okay? For that reason, muscle cells are often referred to as muscle fibers. So I'll probably use that term throughout the rest of this lecture. When I say muscle fiber, I'm just referring to a muscle cell, okay? The length of that fiber can vary from very short to the muscles in your eye, to very long, such as the muscles in your legs, okay? And really the reason they have that shape is because of their function. Their function is to transmit force and pull on bones. A circular cell would not really be able to do that, so the cylindrical shape is very much appropriate based upon the function of a muscle, which is to produce movement. As we'll see in a little bit, those fibers can shorten, and that produces flexion, and then they can lengthen, and that allows for extension and relaxation, okay? So let's look at the structure a little bit more closely. What we see here on the left side of the screen is what is known as a muscle fiber, okay? So notice within a muscle fiber, there are smaller cylindrical organelles as well, which we'll talk about here shortly, okay? And this is perhaps a better picture of the muscle fiber. So muscle fibers are wrapped in what is known as the sarcolemma, which is kind of like, the sarcolemma is the cell membrane of a muscle fiber, okay? So individual muscle fibers are wrapped in a sarcolemma, but then bundles of muscle fibers are wrapped in what are known as fascicles, okay? A fascicle just refers to a bundle of muscle fibers, okay? And then the fascicles, or the fasciculi, the proper way to refer to the plural, are then bundled together into muscles, right? So a muscle is a bundle of fascicles. A fascicle is a bundle of muscle fibers. So we sort of have these like this larger unit that gets progressively smaller and smaller and smaller into these individual functional units, right? And a muscle fiber would be like an individual functional unit, okay? And the other thing I want to point out on this slide is, let's see, we have these different layers of connective tissue, the epimysium, perimysium, endomysium. The endomysium wraps individual muscle fibers. The paramysium will wrap fasciculi, and the epimysium is basically wraps the entire muscle. So we have all of these connective tissue layers, but an important thing to point out is that all of these connective tissue layers that run deep within a muscle fiber all the way to the outer layer of the muscle itself, they all fuse together into a tendon and attach to the bone, right? So not only is the muscle as a whole attached to the bone, but each individual muscle fiber is also attached to the bone. And that allows for the tremendous amount of force production that when all of our muscle fibers join together to shorten, all of that force generated from, that's sort of like accumulated from every individual muscle fiber, transmits force onto that bone and produces movement, okay? Notice one other thing on this picture. We have mitochondria that are sort of spread throughout the muscle fiber. And that's important because, you know, as we'll learn, all of these individual proteins, which are shown here, they all are going to need ATP. So having mitochondria spread throughout the muscle fiber allows for ATP to be sort of on demand for all of these different contractile proteins. The other thing is you can see here there's a nucleus, and then here there's a nucleus. What's not shown in this picture is that there would be several nuclei, and when I say several, like hundreds, right? Most cells just have one nucleus, and that nucleus is usually like in the middle of the cell. Because a muscle fiber is so long, it's called multinucleated, which means it has multiple nuclei, several nucleuses, which is not really a word. And that has a few very important functions that we're not going to get into in this class, but, you know, simply a nucleus kind of controls the space around it. In a normal body cell, the space around the nucleus, which would be in the middle, is pretty tight, but a muscle fiber is very long. So there has to be a lot of nuclei present because that cell can potentially be so much larger than a typical circular body cell. So this slide just goes through sort of the layers of organization that I mentioned earlier. So take a minute to pause the recording, read what the definition of each of these terms is. And again, these are just the layers of connective tissue that are incorporated in skeletal muscle. There's a question I'll ask you on the lecture study questions. What is the name of the cell membrane of a muscle fiber? That is called the sarcolemma, okay? And then normal body cells, the fluid inside of it is called cytoplasm. In a muscle fiber, it's called sarcoplasm. Sarco is a word that, like a Greek word meaning flesh or muscle. So that's why you see the prefix sarco used in a lot of words that refer to skeletal muscle anatomy and physiology. So within the sarcoplasm is where you have mitochondria, the DNA, the nuclei, I should say, that house the DNA, and then there's glycogen stored in the sarcoplasm and other specialized organelles, okay? And then the other thing we'll talk about here in a second is the sarcoplasmic reticulum, which is essentially a network of channels that allows for the distribution of calcium throughout the muscle fiber. And we'll talk about the role of calcium a little bit later, but let's jump back real quick and notice the sarcoplasmic reticulum is shown here. So it's like a network of tubes that transverses the whole length of a muscle fiber. And again, this is going to ultimately allow for calcium to be funneled into the entire muscle fiber. Calcium has a very important role in muscle contraction, as we'll learn when we get to the physiology part. So as I mentioned earlier, muscle force is transmitted directly to a bone from the connective tissue, and then ultimately the tendons. These terms I'm not super worried about, but it just tells you the terms used to describe the points where a muscle connects to a bone. You have the origin, which is where the tendon is attached, generally at the proximal end of a bone, so farther away. And then the insertion is, the proximal is the closer end. So if we're looking at the humerus, proximal is closer to the midline of the body, so that would be at this end. And the distal end would be more distant from the midline, so the distal end would be near the elbow, right? So the origin of a muscle is usually at the proximal end. The insertion of a muscle is usually at the distal end. The insertion is the point where movement would occur. So the insertion of my biceps muscle would be here. When my biceps contracts, that's going to produce movement at the distal end of the humerus bone. So what are muscles made of? Mostly water and protein, okay? As our body in general is composed of, the muscles themselves are roughly 75% water, okay? About 20% protein. So there are key proteins that are known as contractile proteins. These are the most abundant proteins in a muscle, which makes sense because contractile proteins are what produce movement. The major role of muscles is to produce movement, okay? So here are the three key contractile proteins that we'll talk about shortly, okay? And then there are other substances found in a muscle. Salts, I mentioned carbohydrate. That makes up about 5% of an individual muscle fiber. So going back to the structure, perhaps the most important organelle in a skeletal muscle fiber is what is known as the myofibril. So I'm going to jump back to my picture real quick. Actually, we could just go ahead and we'll come back to that information slide here in just a second. But if we said this was a muscle fiber, right? Within a muscle fiber, there are several cylindrical, what we'll just call for now, organelles called myofibrils. And that is represented by this part of the picture. So notice within this muscle fiber, there are 8 myofibrils bundled together, okay? Myofibrils are made up of a series of what are called sarcomeres, which is shown here, right? So in this myofibril here, we have a sarcomere here, we have one here, here, here. Sarcomeres are divided by this zigzag blue line, which we'll identify in just a moment. Sarcomeres then are composed of these different contractile proteins, which we see described here. But let me jump back to the information slide just a second. Make sure I'm not getting ahead of myself, okay? So the myofibrils contain smaller subunits, again called the sarcomeres, which are composed of what are known as the myofilaments. The myofilaments refer to these contractile proteins, okay? The two major contractile proteins, or myofilaments, are actin and myosin, and these make up about 85% of this myofibrillar complex, as they refer to it there, okay? There are some other myofilaments as well that have lesser importance, and those are listed at the bottom. Now, as we'll see, these are more, they add support and they have more of a structural function, where the actin and myosin are the two that facilitate the contraction. These other proteins sort of have a support role for actin and myosin. Let's jump back to our picture and take a look at where these are. So if we pull out an individual sarcomere, which again, a sarcomere is pictured here. A myofibril consists of a series of sarcomeres laid end to end, okay? A lot of people refer to the sarcomere as the individual functional unit of a skeletal muscle. So it is these sarcomeres that shorten and lengthen individually, but they also occur collectively as well. So all of these sarcomeres are going to shorten and lengthen at the same time. And when all of them shorten, the muscle, in the term of the bicep, would contract and produce movement. And when they all lengthen or relax, that allows for the extension or the relaxation of the muscle, okay? Let's take a look at the sarcomere. So the sarcomere mainly is where the two major contractile proteins are housed. We have the myosin, which we refer to as the thick filament because it is quite a bit thicker, right? And that is shown here, here, here. Sorry. And then we have the actin, which is the thin filament, which is shown in blue, and that is found here, 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 and here, okay? So these overlap with one another, and on the myosin filaments are a series of what are known as these cross bridges, which are shown here. Cross bridges kind of look like little golf club heads, okay? Now, these golf club heads, these cross bridges are attached to the actin, okay? When they are energized with ATP, these golf club heads will sort of cycle, right? They'll flip inward, producing shortening of the entire sarcomere. So not all of these, but a percentage of the cross bridges are going to cycle at one time, and that's going to bring the whole sarcomere closer together. And if you think about it, if all of the sarcomeres within this myofibril and then even within this entire muscle, if they all shorten at the same time, that's going to make the entire muscle shorten, and that's going to produce movement, okay? A new ATP molecule will have to move in and allow the cross-bridge head to relax, okay? And when that occurs throughout an entire muscle, the muscle will relax and it would lengthen out, okay? Now we're going to get into more of the physiology here shortly, but it's kind of impossible to talk about the anatomy without throwing in some physiology at the same time. If we come back down, dialed in, this is sort of a picture using an electron myograph microscope that shows what it actually looks like inside the body. So notice in the middle of the sarcomere, we have this thing called the M band. The M band is more of a stabilizing protein that keeps these myosin proteins in place, okay? Then on the outer edges of the sarcomere, we have what is known as the Z line or the Z disc. The Z disc is what the actin molecules are anchored to, okay? So again, I said there were structural proteins, and that's what these kind of represent. On the actin filament, we also have two smaller regulatory proteins, the tropomyosin, which is sort of this really thin, where's my arrow? There it is. This really thin rope-like protein here. This has a very important role in function. And then on top of that, we have these troponin proteins, which are shown here, 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 and here. So again, these have a very important role with regard to the function, and we'll talk about that here in just a little bit. Let me jump back, make sure I didn't miss anything on this slide. The M protein, remember that's what is anchored in the middle. That's what anchors the myosin, and then the Z disc is just basically a connective tissue layer, as you can see here. It separates each individual sarcomeres, but that's what the actin is bound to, right? So when these actin are attached to the myosin, when there is contraction of the myosin cross-bridge heads, the entire sarcomere would shorten, and in turn, the entire muscle fiber and muscle would shorten as well. Now, all of these bands are labeled. Personally, I don't put too much importance on these things here, but what the A band refers to the zone where the myosin is found, and the I band refers to the zone where there is no myosin. So notice here on this picture, like the A band would be in this zone here, which usually appears darker or more red in color. Like here, you can see the A band pretty well. The I band would be the portion of the myofibril where there is no myosin, so it usually appears lighter in color. So it would be represented by this area here, here, and in the picture at the bottom, it would be this area here. If we jump up to the myofibril, the A band is here. The I band actually sort of spreads out across two consecutive sarcomeres because it represents the area where there's no myosin. Okay. Again, I don't put too much importance on those terms, but of course I want you all to be aware of what they mean. Okay. Now, what I do want to note out one more thing on the Z disc. Oops, wrong way. The Z disc is, again, where the actin is attached. The Z disc is actually then attached to the overall cell membrane, which is then attached ultimately to the bone. So the connective tissue at the individual sarcomere unit is itself attached ultimately to the tendon, which again shows us the importance of the contraction of each individual sarcomere has the ability to contribute to force production and movement of a bone and the production of force by a specific muscle. Okay. Now, this is maybe a better picture that shows what this looks like. I've already sort of gone through this in detail. So you might want to just take a moment, pause your recording, read these bullet points, and refer to the picture. Again, I'll point out a couple things before we move on. Now, notice in this picture, it looks like there's only, that the sarcomeres are, that the myosin proteins are two-dimensional, but it's actually three-dimensional. So we'll get a better look at this later as well. But notice there are a ton of these cross bridges, right? It's not like there's just one on this side, one on this side. It's a three-dimensional object, okay? And they don't all attach at the same time, right? If we're moving something light, like I'm picking my phone up, I might not need all of my cross bridges to contribute to force production to move this phone. But if I pick up like a 50-pound dumbbell, I'm going to need more of my cross bridges, so more of them will attach, right? We have the ability to grade force production by controlling how many of these cross bridges are attached and contributing to force production at a given time. There's no reason for all of these to attach and move for me to just pick this phone up, right? The body's going to be more efficient and really save energy because the movement of each individual cross bridge is going to use an ATP molecule. So there's no reason to use a ton of ATP molecules just to move this phone, right? It's not light. It is light. So I don't need all of my cross bridges to connect and contract. What else on this slide? So let's see. The Z line is composed of this other protein, A-actin, that I showed on the previous slide. Nebulin is just a support protein, right? So there are these super thin proteins that help really just keep the structure together and prohibit a lot of like lateral and up and down movement within the sarcomere. The M band is composed of these different M proteins. And then again, we have these C, X, and H proteins. Gosh, sorry that keeps happening. Which again, just provides us with some framework really just to keep the sarcomere nice and uniform and limit any sort of lateral up and down or three-dimensional movement to really allow for all of the force production to be transmitted in this direction. Now, I did mention this earlier that this is a three-dimensional. So if you just look at this picture, it almost looks like that it's more of a two-dimensional situation, but that's not the case. So if you look here, this gives you kind of a better picture that each individual myosin is surrounded by actin, right? So the cross bridges actually encircle the filament, and it can be attached, in this picture at least, in 6 points, right? So each individual myosin is surrounded by 6 individual actins, which again is going to facilitate greater force production because that myosin is surrounded by actin. It's not like it's just two-dimensional and it can just attach here and here. It's more three-dimensional where it can attach and pull, release. That's going to allow for greater force production capabilities. So look at this. In a single fiber, there are approximately 16 billion myosin, which are the thick filaments, and there's a 6-to-1 ratio. So there's 64 billion thin filaments in an individual muscle fiber, myofibril, I should say. This gives you a better look at the cross-bridge heads, okay? And this is where we're going to get a little bit more into the physiology. That notice that we have these sites that are called the active sites. That's where the myosin attaches to the actin. Now, in a relaxed muscle, that active site is going to be blocked by the tropomyosin. So remember, the tropomyosin is this super thin yellow line. This prohibits muscle contraction in a relaxed muscle, okay? On top of the tropomyosin, we have this other protein that I mentioned earlier called troponin, okay? Ultimately, as we'll see, calcium is going to move into a muscle fiber when it is activated, stimulated from the nervous system. Calcium is going to bind to troponin. Troponin is then going to change its shape. And when it does that, it's going to pull the tropomyosin out of the way and expose the active site, which then allows the myosin to bind to the actin, right? So there's a lot of things that have to occur prior to muscle contraction, and we'll get into that here in just a little bit. But I just wanted you to get a better look at the myosin heads. Now, the myosin head is important because this is where ATP is going to bind to. We talked a lot about the production of ATP, what its role is. Ultimately, ATP is going to exist to allow these myosin heads to move. So the energy liberated from the ATP energizes this myosin head and allows it to move. So if your body's not producing ATP, you're not going to be able to move, right? So when you die, your muscles don't work because your body's no longer making energy. That's a very simple explanation, but you get the point, right? ATP hydrolysis is the word that is often used for the splitting apart of ATP, liberating the energy, which, as it says here, then is used to activate the myosin's two heads. So notice there are two located per filament. When they're able to attach, they automatically cycle, causing the entire muscle to shorten. Now, I did mention calcium on the last slide. That, jumping back to what I mentioned earlier, the purpose of these, the sarcoplasmic reticulum is to allow calcium to be delivered to the entire muscle fiber, right? If each individual troponin is going to need calcium, there's going to be a ton of calcium that's required for a muscle to contract, right? So calcium is stored in what are known as the T-tubules, okay? But then, it's stored in the sarcoplasmic reticulum. And when muscle contraction is imminent, the calcium is released from these things here called the T-tubules. So the T-tubules sort of make the boundaries for the sarcoplasmic reticulum. And again, that's going to allow for widespread delivery of calcium when muscle contraction needs to occur. We'll get into that in a little bit more detail here in just a little bit. Why don't we do that now? Okay. So there's a theory that has been developed that explains muscle contraction. Okay. This theory was developed over 60 years ago. It's undergone some changes. Even in the 20 years I've been studying this, there's been a little bit of modification, but it's essentially the same, okay? But it's important to note that, right? This is a theory, right? So that doesn't mean it's the gospel. It doesn't mean this is the final decision on how this works. It means we're still learning about these things. And if we learn something new, the theory will be modified, all right? Most of science is a theory, right? This is the best idea we have of how it works. We shouldn't ever think that anything told by science is the absolute last word on it, unless it's something that we can really fully investigate to the best detail. Because this stuff is so small, it's really hard to know exactly how it works, but this is the theory of how we think it works. Just remember that as you continue your studies. Don't ever be afraid to challenge theories, because that's how we continue to learn more details, okay? Okay, so what this theory is called is the sliding filament theory model, and this is kind of what I've been referring to off and on throughout this presentation thus far, is that muscle shortening and lengthening occurs because these filaments either slide closer together or they spread apart, right? These filaments slide past one another, kind of like my fingers are doing here. And when they come together, that produces muscle shortening, which is contraction. And then when they slide apart, that is muscle lengthening, which is relaxation, okay? So these myosin cross bridges cyclically attach, rotate, and detach, and pull the Z discs closer together during contraction. And then when they detach, all of them detach simultaneously. That allows the Z disc to move back into resting position. So this picture here sort of indicates what a resting sarcomere looks like. Now notice there is some overlap, but there's plenty of room for movement. This at the bottom shows what sort of a maximal contraction looks like, where we have full overlap, indicating every single myosin would be in contact with a actin, okay? This one is actually optimal overlap here. What this one shows would be like an overstretched muscle. So notice here, there is a lot of opportunity for myosin to connect to actin, which would allow for rapid force production. With this one, there's limited interaction. So your ability to produce force is going to be lessened if the muscle is overstretched. So that's why a lot of people have said that really aggressive static stretching prior to lifting weights can actually limit your force production. This kind of offers us a visual of why that can occur. If you're overstretched prior to lifting weights, then you're going to have less than optimal overlap. In order to produce force, you have to have some overlap. This is less than optimal, where this picture gives us a more visual representation of what optimal overlap would look like in terms of actin and myosin. So these pictures show us, again, the role of the cross-bridge heads, actin, but now also our regulatory proteins here, okay? Again, I'll point out that ATP is necessary for this process, that ATP would attach to the myosin, and that the enzyme that we talked about that splits ATP apart, ATPase, is found in high concentrations on these cross-bridge heads. So as soon as an ATP binds, the enzyme is there ready to bust it apart, liberating that energy, and then sort of energizing that cross-bridge head and allowing it to cycle back and forth. This shows us that when the cross-bridge head attaches, it then can cycle and produce movement, right, which would shorten the sarcomere, okay? It can then detach and bind again and continue to cycle. So sort of like these say that we're moving this direction. One cycles, the next cycles. One cycles, the next cycles. They keep cycling back and forth. As long as we're providing that input that we want to move that muscle, these cross bridges are going to continue to cycle. It's asynchronous, which means they do not all attach and cycle at the same time, but they sort of work like one would go, then the next one would go, then this one will relax and go, then this one relax and goes. That allows us to maintain force production. If they all cycled and relaxed at the same time, our force production would sort of be more cyclical, but we can sustain it without any drops because they cycle asynchronously, which means not at the same time. And that point is listed here. They move like oars, knifing through the water, but they do not move asynchronously. It's kind of like when you row a boat. If anybody's ever gone canoeing, right? You put one pedal on this side, then you go to the other side, and you go back and forth. You don't row both at the same time. Typically, if you have like a long oar, you're going one side, one side, one side, one side, one side, one side. That's kind of more similar to how our cross bridges cycle. Only about 50% are in contact at a given time. This kind of speaks to the overlap that I mentioned earlier, that force production is optimal at an optimal sarcomere length, which is going to be shown really like in this picture here, right? We have to have room for them to overlap. If it's too short, we can't, there's no movement, which is obvious. If it's too long, then we can't initiate movement. There has to be some overlap between the actin and myosin for movement to even be initiated, and then for it to be efficient in its force production capabilities. This is referred to as the length-tension relationship. Again, this is not something I'm super worried about you all learning, but I just want to throw it out there today, as I know I'm throwing a lot of information at you today. Here's more of a graphical depiction of the same idea, that force production ability is the highest in this zone here, where we have sarcomere length corresponding to these numbers, right? Same thing here. Force production is the highest at this level of sarcomere length. All right, so what's the link with ATP? I've already talked about this quite a bit, but for cross bridges to detach and attach, each phase would require a molecule of ATP. So really there's 2 ATP required per cycle. One to attach it, and then one to detach it, okay? A good example of this is the phenomenon that is known as rigor mortis, something you all have probably heard of, where when somebody dies, they become very stiff, right? In fact, dead bodies are often referred to as stiffs, okay? Now, where did this come from? Well, according to what I just said, energy is required not only for cross bridges to move, but energy is required for them to relax, right? So if somebody dies, when they die, they're no longer going to produce ATP. Therefore, the cross bridges are going to stay bound to the actin because energy is required for them to detach, okay? That's why immediately after death, people are very stiff. Okay, now eventually, after a certain amount of time, which I don't know how long this takes, the proteins start to degrade and they're able to detach. But in that initial period, when there's no ATP being produced because the person is dead, those cross bridges stay bound to the actin, and that's why they have a very stiff and rigid feel to the body. I know that's kind of morbid, but that helps illustrate the point of ATP is not only required for movement and attachment, but it's also required for detachment. This also shows us why we need to be able to produce so much ATP at all times as well. Myosin ATPase just refers to the form of ATPase that is found on the cross-bridge heads. I mentioned that a second ago, that if we jump back to this picture, here, there's literally ATPase that hangs out on these cross-bridge heads, and that's referred to as myosin ATPase. All right, now the process that leads to the sliding filament theory is known as the excitation contraction coupling theory, okay? Excitation refers to your brain activating the muscles through this process of excitation, right? We're exciting a nerve, and that's going to then lead to contraction. Now we're going to get into the excitation portion of this a little bit more in our last chapter of the year, which is about the nervous system. So we're going to kind of skip part of that today and get to the more skeletal muscle-based stuff. But essentially, when our nervous system is activated, there is an electrical signal that is sent from our brain to a given muscle. And that electrical impulse from our brain stimulates the release of, you guessed it, calcium, right? The excitation is ultimately going to cause the release of calcium into the muscle fiber, and that calcium is then going to allow for contraction to occur. So the excitation phase is going to culminate in the release of calcium into the muscle fiber. That calcium then allows for the contractile process to occur, because calcium, as I mentioned earlier, ultimately allows actin and myosin to bind, and then that can facilitate or will promote the movement that occurs within each of our individual muscles. Don't worry too much about these two boxes. I feel like this is just going to be confusing, and I've never really seen a textbook refer to this, so just don't worry too much about that. Just note really with this slide the importance of calcium. Excitation from the nervous system ends or culminates in the release of calcium. This then allows for the contraction process to occur. Okay. So in a relaxed state, the calcium binding site is here, and the active site is blocked by tropomyosin. When calcium floods into the muscle, though, calcium attaches to troponin. Okay. Troponin changes shape, and then the tropomyosin is moved out of the way, exposing the active site, or what they call here the myosin binding site, and that allows for the cross bridge to bind. So in the relaxed state, no calcium, troponin, tropomyosin, I should say, is blocking the binding site, so it can't attach. Okay. Once calcium moves in, tropomyosin is activated. Troponin is activated. Tropomyosin is pulled out of the way, and that then allows the myosin cross bridge to bind to actin at the myosin binding site. You can see, gosh darn it, my mouse is so sensitive, as you all have noticed this semester. Whenever I move it a certain way, the slides jump ahead. I'm sorry. Each of these actin molecules, let me move slowly so I can show you this, has a binding site that is shown here. Now what you can't see, because it's blocked, is the tropomyosin is covering the binding site here. But once the tropomyosin moves out of the way, the binding site is exposed, allowing the cross bridge to attach right here. Okay. And again, calcium has to be present for the binding site to be exposed and myosin to be able to attach, right? So calcium is critical when it comes to muscle contraction. And really this shows us the importance of us having adequate calcium levels in our body. Not only would depleted calcium have a negative impact on bone health, depleted calcium can have a negative impact on muscle contraction. Now for relaxation, as long as calcium is present in a cell, contraction is going to keep occurring. But as soon as I consciously decide, say I'm flexing, I'm gonna consciously decide to relax this muscle. When that happens, the signal from my brain goes away. Calcium is then removed from the muscle, right? And that then moves tropomyosin back into blocking position and causes the relaxation of this muscle, right? So deactivation from the brain basically prevents any mechanical link between actin and myosin, and it also then inhibits ATPase activity, right? Which would save energy and prohibit any more ATP from being split, because we're now in a relaxation phase. But if I consciously again decide to flex this muscle, calcium is flooding into this muscle, allowing for contraction to occur. Now I'm going to consciously decide to relax it. Now calcium is going to be withdrawn from that muscle and stored back in the sarcoplasmic reticulum. So calcium, very important for muscle contraction. So what we see here are essentially a sequence of steps These two are more based upon what happens in the nervous system, so I'm not going to talk about those today. We kind of picked it up here where once the T-tubule gets stimulated from the nervous system, calcium is released from the sarcoplasmic reticulum. Calcium binds to troponin. Tropomyosin is moved out of the way, which allows actin to combine with myosin and the ATP that is present. The ATP is split by the enzyme, and then that energy that is liberated from the ATP molecule allows for the cross-bridge movement. So this is the excitation contraction process, sort of step 1 through 5, really all of it, where step 5 is more the sliding filament theory. So the sliding filament theory is sort of rolled into the excitation contraction coupling. The sliding filament theory just refers specifically to the movement that occurs with the proteins. This kind of tells us that as long as calcium is present, as well as ATP, that cross-bridge activation will continue. But once we remove the stimulus of this muscle from our nervous system, calcium is sucked back up, sort of vacuumed up back into the sarcoplasmic reticulum. And in the absence of calcium, as long as ATP is present, then the actin and myosin will detach and relax, and tropomyosin will move back into blocking position. Go back, pause the recording, read through these steps, okay, and then come to this picture and sort of follow these same steps are described again here with the visual representation. So I'm not going to read this to you, but I highly recommend, again, pause this recording and read through the steps with the visual, because I think that'll really help you sort of wrap your head around this process. When you're thinking about these physiological things, it's really hard to wrap your head around it unless you can see it. This is the best I can do in terms of showing it to you. You might also, if you're interested, go to YouTube and look up animations of this, because animations will actually help a lot as well. And there's a lot of good ones on YouTube that you could easily find. In fact, maybe I'll try to find some and post them in our folder. I'll do that. Okay, we're going to finish up here. I'm losing my voice, so I need to wrap it up. But muscle fibers are not all the same, right? We have some muscle fibers that allow us to produce explosive force, and some muscle fibers that allow us to sustain low intensity force, right? And we separate those muscle fibers into types, okay? The type is influenced by a lot of factors, okay? One factor is what is the main mechanism for the production of ATP. Low intensity, low force production fibers, they typically are going to use aerobic metabolism, where explosive force production fibers, they're going to use anaerobic, okay? What type of motor neuron innervates them? Very fast motor neurons are required for those explosive fibers. Slower motor neurons are required for the endurance-related fibers, okay? What type of myosin is present? High force production fibers are going to have a different type of myosin versus low force production, okay? So there's a lot of factors, as we'll see, that influence the fiber type, okay? This is a good chart that shows you, and you all are going to need to use this slide a lot in the study questions and in the critical thinking assignment, okay? This shows you the major fiber types in the human body. Now, type 2B is more of a controversial fiber. When I was a student, we had type 2A and type 2B. It is now thought that humans have very little type 2B. These are more found in animals. Instead, we have what is known as type 2X, okay? So we have our global type 1 and type 2, but type 2 can be divided into two subtypes, type 2A, type 2X. So if we notice this chart, we're gonna contrast type 1 and type 2X because these are the most glaring opposites. Contraction time for type 1 is slow. Contraction time for type 2X is fast. Size of motor neuron, type 1 small, large. Resistance to fatigue is high because it uses the aerobic energy system. Resistance to fatigue in type 2X is, it says intermediate, but it's kind of low because it uses short-term anaerobic. Okay. Maximum duration for use of type 1, many hours because it uses aerobic. Maximum duration for type 2X, less than 5 minutes because it uses short-term anaerobic, right? So the energy system that the fiber type relies on primarily is a really key component, and it determines the characteristics of the muscle fiber, right? Because type 1 relies on aerobic metabolism, it's going to have a very high concentration of mitochondria. It's going to have a lot of blood vessels surrounding it to facilitate getting oxygen into the fiber. On the other hand, low to medium capillary density and mitochondrial density for type 2X because they rely mostly on, again, your anaerobic energy system, okay? Low glycogen capacity in type 1 because they rely on mostly fat for fuel. High glycogen capacity in type 2X because they're going to rely mostly on carbohydrate for fuel, okay? Excuse me. So in these next few slides, it's going to break down sort of the main characteristics of each of the fiber types. So for your critical thinking questions, I'm going to ask you to list at least 3 or 4 characteristics of one of the fiber types. I can't remember which one, okay? But then for your critical thinking assignment, I'm going to ask you to pick a sport or an activity. It could be playing basketball. It could be running a marathon. It could be doing one single broad jump. I don't care what you pick. Pick an activity. Tell me what the activity is, okay? Then I want you to use this chart and think about, based upon how you perform that activity, which of these muscle fibers is going to be the one that mainly contributes to that activity. Now, you're never going to just use one. Even sprinting a short distance, even though it's going to rely mostly on type 2X, type 1 fibers are still going to be used, but it's primarily going to rely on this. On the opposite end of the spectrum, a marathon 26-mile run is mostly going to use type 1, but there's still going to be some contribution from type 2X. So I want you to think about which fiber type is the predominant one used for the activity that you pick, okay? And then I'm going to ask you to explain a few reasons why, right? For example, for a marathon, because the duration of the activity is so long, we're going to need to rely on our aerobic energy system to get us through that activity, okay? We're going to need a lot of mitochondria to get through that activity. So we need high mitochondrial density. We're going to need a lot of oxygen-rich blood to get to the muscles, so we're going to need high capillary density, okay? We're going to need to sustain energy production for a long period of time, so we're going to have to use fat for fuel, right? Which the mitochondria can do. So when you're answering the critical thinking questions, sort of use what I just used as an example, use this chart as a guide, think about the fiber type that primarily fuels the activity or primary fiber type that is used in that activity, and then give me some, I think I asked for 3 specific examples of how the characteristics of that fiber type relate to the ability to conduct or participate in that activity, okay? You can use the next few slides to help you as well. These sort of summarize the highlights of the chart, so I'm not going to talk about these next few slides specifically, but when you're answering your why questions, you might refer to these slides. Once you figure out the fiber type you use for the activity, you can use these few slides here to get sort of the high points of those muscle fibers, okay? Let me just go back and say one thing before I wrap it up. Type 2A is sort of like a hybrid fiber. Type 2A has characteristics of both type 1 and type 2X, right? So a lot of people think type 2A is kind of like the super fiber because it has good force production, but it also has good resistance to fatigue, right? It's sort of the happy medium between the two fiber types. And honestly, if you are somebody who trains in sort of a well-rounded way, you do both endurance activity and explosive resistance training, sprinting, etc., you're going to really develop these types of muscle fibers. Muscle fibers are not set in stone. They're plastic. They have plasticity to them, which means they will adapt to how you train. If you train only doing slow aerobic activity, you're going to sway more this direction. If you train only doing high intensity, short duration activity, you're going to go more this direction. If you train using both, you're going to sort of be in the middle, which I think personally, this is the most, the human with the most flexibility for doing different types of activities. You want to have a higher percentage of type 2A fibers. This just shows us how muscle fibers are extracted. This is a very painful process. So if you sign up for a research study that uses muscle fiber typing, be wary that the process might hurt a little bit. This is what is known as a biopsy, where they inject this needle and they extract a chunk of your muscle fibers, and they would then look at them under a microscope to see what your percentage of different muscle fiber types are. Notice that the muscle fibers are grouped together sort of sporadically, which is good. There is some genetic predisposition to certain fiber types or the other. You probably recognize this in your own life, maybe within the lives of your parents, that if you're good at cardio, you're going to have more type 1 muscle fibers. If you're more of an explosive athlete that can run fast and jump, you're going to have more type 2 fibers. Usually it's roughly 50-50, but you're going to sway slightly one way or the other, and that's going to be based upon your genetics as well as your training as well, okay? This kind of just shows us that with different sports that there are different percentage. So somebody who's good at extreme long distance events, they're going to have mostly type 1, which was slow twitch, type 1. People who are explosive, they're going to have less type 1, slow twitch, more type 2, fast twitch. Okay, so I'm about out of voice, so I'm going to stop. Study questions, and we're going to go back to a critical thinking assignment for this chapter, which again is going to ask you to think about an activity and tell me which fiber type is going to be the one that is used most for that activity and why. Okay, so that's it. I'll talk to you all soon. Bye.